Hello everyone, welcome to Chapter 4 video lecture. I'll be discussing energy and cellular metabolism. Alright, here are the learning objectives for this chapter. So we're going to spend a lot of time talking about chemical reactions before we get into how cells extract energy from certain molecules. So we'll first just start off defining what energy is, and how that is used to accomplish work, what types of different work there is, as well as the energy. We'll get into a little bit of physics here, just very briefly on two laws as they relate to thermodynamics and the universe, because even though you're in a physiology class, we still have to abide by universal laws, universal chemical principles. After we establish some energy principles, we're going to move on talking about chemical reactions, so free energy, endergonic, exergonic reactions, and we're going to apply that to how some reactions are what we call reversible and some are irreversible. This will then get us into the concept of enzymes and how they assist or facilitate biological reactions, and then we'll define a few different types of enzymatic reactions, chemical reactions that we're going to encounter not only in this chapter, but for the remainder of this class. And then after that, we transition into metabolism. So I'll introduce you to the concepts of what metabolism versus anabolism and catabolism are, how cells control the flow of molecules, how they, I should say, regulate metabolism. And then we'll start getting into metabolic pathways and molecules that are involved in those pathways, such as ADP, ADP, FADH2, and NADH. And then this learning objective here, 4.4.4, if I said that correctly, it's a little misleading, I think, because it says outline the pathways for aerobic and anaerobic metabolism of glucose. We're really just getting, what that means is we're going to go over three different metabolic pathways: glycolysis, citric acid cycle, or TCA cycle, or Krebs cycle, depending on your preferred term, as well as the electron transport chain. So so that's a big learning objective, and it's kind of vague and doesn't really indicate just how much material is going to apply to that one. And then we'll finish up the chapter with discussing how the electron transport system, electron transport chains create those, recreate the high energy bonds of ATP. Okay, so here's the overview for the chapter. We're going to start off discussing those energy in biological systems, move on to chemical reactions, discuss some enzymes, and then we'll finish it up with metabolism. Okay, so we have to define what energy is and why our cells need it. Point is, so when we say energy, cells need energy. What does that really mean? They need that energy to perform work. Okay? So energy is defined as the capacity to do work in biological systems. But when we're talking about biological systems, there are three types of work that we can refer to. So there's chemical work, transport work, and mechanical work. This chapter is going to focus on chemical work, making and breaking of chemical bonds. But however, very quickly, once we get into chapters 5 and 6, we're going to talk a lot about transport work, how cells allow or move ions across the cell membrane, and could also be within the cell itself. And then another chapter is going to, well, later on in the semester, we'll talk a lot about mechanical work. This is work that's used for movement, and this can be on the microscopic or macroscopic level. So sometimes we need to move some organelles or vesicles within a cell. We need to transport them around. That's mechanical work. However, you're probably thinking mechanical work and muscle contraction. That's a macroscopic level, how we actually contract our muscles through the use of motor proteins. Okay, so right now, let's just talk a little bit more about this first type of work, chemical work. Okay. Another thing to realize about energy, yes, we need it to do work, But that energy itself comes in two different forms, and you should probably have encountered this in previous classes if you took maybe if you took physics in high school, or if you've taken it before here at Gonzaga. So two forms of energy we're going to briefly cover is kinetic. So this is energy that is in motion. Okay, so right now I'm speaking to you through this video. That is me talking. That's moving my mouth. That's kinetic. I'm using kinetic energy to accomplish that. Also, I'm waving my hands around because that's what people do when they talk. That is kinetic energy. I have an example here, a ball rolling down a hill. So something that's in motion, that is an example of kinetic energy. In a molecular sense, so molecules moving across a cell membrane, that's also a form of kinetic energy. However, energy doesn't have to be in motion all the time. It can be, I don't say sitting still, but it can be non-moving as well. And that form of non-moving energy is what we call potential energy, aka stored energy. So instead of a ball rolling down a hill, well, that ball, when it's poised at the top of the hill, it's not moving yet, but it has the potential to roll down that hill. That's what we call potential energy. And then again, in a chemical or biological sense, when we talk, this chapter we're going to talk a lot about chemical bonds and making them or breaking them. And the reason is chemical bonds are stored energy. Okay? So they have the potential, when those bonds are broken, they release energy. So that's what we mean by potential energy being stored in chemical bonds. That's a really critical key point to understand before moving on to the rest of this material in this chapter. And then another thing to know about energy is it can be converted. Okay? So as that ball, so going back to the ball example, as it's poised at the top of the hill, it's sitting there, it's not in motion, that's potential. But as soon as it starts to move for whatever reason, then that energy is converted into kinetic energy. So that's a little bit on energy, how it's used to perform work, and a couple different types of energy. However, let's, instead of using a ball and a non-biological example, all living organisms that we're aware of have to obtain energy in order to survive, in order to provide the energy for their cells to do some form of work, chemical, mechanical, or transport work. Okay? So in the case of the human body, we obviously have to eat. We have to obtain our energy from our food. And this comes in the form of our macromolecules, carbs, lipids, and protein. Those are our sources of energy. Plants, they use the sun that the energy provides in order to make glucose or carbohydrates, therefore their source of energy. Okay? So here's just a diagram illustrating that concept of kinetic potential energy and how the energy can be converted from one form to another. I feel like this is pretty straightforward. So I guess this diagram is also illustrating that whether that movement is either up the hill or down the hill, as long as it's in motion, it's still kinetic energy. The only time in this example we'd have potential energy is when the ball is sitting at the top of this ramp and it's not moving. Okay, so that's a little background into energy and work. Now we have to just establish a little bit of physical laws. Don't worry, we're not going into a deep dive here because everything that we learn in physiology is based on laws in the universe. So this gets us to thermodynamics. And this is the study of how energy is used. I just want you to be aware of two basic rules that govern the transfer of energy in the universe, and this still applies to human body and our individual cells as well. So the first one, hopefully you've encountered this before, and if not, don't worry. Pretty simple here. The first law of thermodynamics, also known as the law of conservation of energy, and what this means is this right here. The amount of energy that is present in our universe is constant. Okay? The universe, as best to our knowledge, is a closed system. Nothing is entering or leaving our universe. And what this also means, why we say it's the energy is constant, is because we're not creating or destroying energy. The amount that is there, and it's not going anywhere. So going back to that concept of converting energy can be converted, but it never increases or decreases. But what I think a better word or term for increase or decrease, you do not destroy or create new energy. You only convert it. Okay? Hopefully that makes sense. The other thing I want to point out is while the universe may be a closed system, the human body and the earth is not a closed system. Okay? So the earth constantly gains energy because it is receiving energy from the sun, and the earth is constantly losing energy, okay, in the form of heat and other things. Same concept applies to the human body. It would be great if we were a closed system because that means we would neither have to eat to take in energy, nor would we lose energy. Okay? We would just be like a battery that constantly stays charged, never gets depleted, you never have to recharge it. That'd be great, but we aren't that way. We have to constantly take in food so that our cells can take that energy, convert it into different form of energy that they can use to perform work. And then we are constantly losing energy in the form of heat because these reactions that we'll talk about this semester, they're not 100% efficient. And because of that, we lose heat, and that's what it means by we are not a closed system. Okay, so that's first law of thermodynamics. Second one is what we call, I think, or I always attribute this to, the way I just remember it is, second law is entropy. Okay, and if you can remember what entropy is, then you know the second law. Okay, so this second law says that natural spontaneous processes always move from a state of higher order to lower order. Okay, or I'd say order and maybe organization might be a better way to think of it. A state of higher organization to a state of lower order. Something is always moving, or what I mean, the second law thing says that things move towards randomness. Okay. This concept is what we call entropy. So when you're creating more randomness, when you're creating more chaos, that's another word you'll probably hear me use when I refer to the second law of thermodynamics. You're creating more chaos, okay? You're creating more entropy. If you want to decrease the entropy in a system, like a human, then it requires the input of energy. So when we eat, to obtain more energy, we're decreasing the entropy temporarily. But as soon as our body starts to use that energy that you've taken in, and it's going to move towards a state of lower order, it's going to create more entropy. Okay, so it's really like a back and forth. And then the other thing to keep in mind about this second law and how it applies not just to the human body, but to the universe, is that based on our current understanding, the universe is always moving towards higher entropy. It's always creating more chaos. Okay, so that's a little background in energy, work, and a little bit on physics and thermodynamics. Now let's shift gears and apply this to chemical reactions. So as I said before, living organisms have to extract energy from their environment in order to survive. So the field of study that focuses on this concept is what we call bioenergetics, and that can be applied to humans, animals, as well as plants, because plants have to obtain energy as well. You can also study bioenergetics on the level of not just a cell, but all the way up to the entire ecosystem, how energy moves through an entire ecosystem. Okay? And at the heart of bioenergetics is this second point right here, how chemical reactions are critical for the transfer of energy. Because remember, we're not creating or destroying new energy, we're only converting it. Bioenergetics is that study of how energy flows and is converted in one form to another. So when we talk about converting or transferring energy, converting it in one form to another through the use of chemical reactions, really what we're getting at is the breaking or making of chemical bonds. Okay? We're either going to release energy or we're going to store energy when we start altering chemical bonds. And again, another kind of re-emphasizing this point here, chemical bonds, when you form a chemical bond, it is a form of potential energy. The other thing I want to say is that hopefully you've learned in some chemistry classes previous to this, there are different types of bonds. There's phosphodiester bonds, there's carboxyl bonds, so there are different bonds present depending on which molecules are being bonded together. And what this means, or what I'm just trying to emphasize here, is that not all bonds are created equal in the sense of the potential energy that they store. Some are higher energy bonds, some are lower energy bonds, and we'll talk about that here in a little bit. Okay, so we now know that making or breaking bonds allows us to transfer or convert energy in our cells. So let's do a deeper dive into some of the types of chemical reactions that are behind these making or breaking of bonds. Okay. Whenever our cells are going to use that potential energy for work, okay, and we have a bond and it's broken, it's going to release energy. Okay, that's called the free energy. So the potential energy that is stored in the bond, once that bond is broken and that energy is released, it's called free energy. Another, kind of to emphasize this, a larger molecule, because it has more bonds, they tend to have larger free energies. And this is where these two compounds over on the right hand of the screen come into play. So we've got glucose up here, and then it's not labeled, but this is an ethanol molecule. Both of these are biomolecules. We have some carbon-carbon bonds, we have some carbon-hydrogen bonds in the ethanol. We have multiple bonds up here. So what I'm getting at, you can hopefully appreciate that glucose is a larger molecule. It has a lot more bonds compared to ethanol. And because of that, you would probably want to assume that glucose has a larger potential energy, a larger free energy that is released once these bonds start getting broken. Okay? But backing up a little bit, these reactions, whether we're breaking or making bonds, they typically require a little bit of input to get them going. It's like a kickstart. If most lawnmowers or weed eaters, you've got to pull that chain, so you've got to spend a little bit of energy before you can start mowing a lawn or weed eating. That's maybe like how I think about this concept of activation energy. So when I'm talking about activation energy, it's defined as the energy and little a for activation. It's a little backwards, okay? So just be aware of that moving forward on the slides. Activation energy is what I'll say is the E of A. Okay? And the book uses an example of what it calls the push up the hill, and that's a really good example as well. And you can imagine that if a little bit of energy is required to start a reaction, then it can come in a range. So a low E of A means that there's a small input of energy that's needed to begin this reaction, and that increases the likelihood that this reaction will occur more often or more easily, aka its chances for occurring spontaneously. On the other end of this, that push up the hill might be a little bit higher. You have to input a little bit more work, energy to get that reaction started. And the higher the activation energy, the higher the likelihood that that reaction will not occur spontaneously. Okay? So this is probably very abstract to you, so let's just look at this in a hill example. So we've got the starting point. We've got an individual right here at the bottom of the hill. He needs to get this boulder over the hill so it can roll down, and as it's rolling down, it's going to be broken up into smaller pieces. So this is the reactant, and the products are what's going to be left at the end of this rolling down the hill, aka this reaction. However, this poor soul has to push this big boulder up the hill, and at this point where he no longer has to start pushing, okay, and the ball starts rolling on itself, this is the chemical reaction. Okay, but this point from here to here, okay, what's being denoted in red, this from here to here, that amount of energy he spends to get up the hill, that would be the activation energy. Okay, so if I were to redraw this hill, this is a different color, and let's say he only had to make it up to here, you can imagine that since that hill is lower, that activation energy that's required to get to that point in this blue hill is lower. So it'd be a lower E of A. At the end of these chemical reactions, I've mentioned, I've alluded to that they can either store or release energy, and that is true. And regardless of whether they store or release energy, we call that a change in free energy as a result of the reaction. Okay. So I guess maybe a more simplified way to think about this is, let's just forget about free energy for right now, and let's just talk about reactions. They either release or store energy, and if a reaction releases energy, then it is what we call an exergonic reaction. Okay? When the free energy, and again, I'll show a diagram of these two, exergonic and endergonic, side by side on the next slide, but just bear with me for a minute. So an exergonic reaction, at the end of the reaction, it's released energy, and the energy level of the products is lower than the free energy of the reactants. Okay? So when you end up with a lower amount of energy, a negative delta G, that means it's an exergonic reaction. An example of this is hydrolyzing or breaking ATP, breaking off one of those phosphodiester bonds and releasing the energy in order to do work, and that's an exergonic reaction. On the opposite side of this, we have endergonic reactions. This is a positive change in G, or free energy. I don't think I mentioned that, but G is free energy, and why it's G is gives free energy for the individual who first described this concept. So endergonic reactions, the free energy at the end of the reaction is higher than the reactants. Okay? So endergonic reactions, they trap, or another way to think of this is they store energy. Okay? The opposite. So on the previous example, I used converting ATP into ADP. So an opposite form of this would be taking that ADP and giving back energy in order to create ATP. Okay, I think this is getting really complex, and really, if I had to summarize both of this, typically an exergonic reaction, you're going to break chemical bonds, and in an endergonic, you're most likely going to make chemical bonds. Okay, that's a real simplified way to look at it. Let's look at this in a diagram. So we've got on the left side here, first up, we've got an exergonic reaction. Okay, remember, exergonic they release energy. So we start out here, the reactants, A plus B, and we still have a hill because almost all chemical reactions require some form of energy to be inputted first. So going up this hill, we've got an activation energy, an E of A, but we're really not worried about that currently. But we get to this top of the hill, the reaction proceeds, and notice, over here on the y-axis, this is the free energy. So we've got energy here, time on the x-axis, energy on the y. Once this reaction starts and it moves forward and it ends, we started here, we ended here. So there's a big difference here. So this is higher. This is lower. It's a negative delta G, negative change in G, and that means it's an endergonic reaction. And in the process, we have released energy, and that released energy will be harnessed to do some form of work, chemical, probably not chemical, probably transport or mechanical. Okay? On the opposite side of this, okay, is that endergonic reaction, a positive delta G, positive Gibbs free energy. So again, we've got this reaction, we're starting here at this energy level. We've got smaller activation energy here, but notice once this reaction proceeds, it ends right here. Okay? It ended higher. It ended higher than where it began, okay, in terms of the y-axis. Okay? So we've trapped or stored some energy in this process. Because of that, it's a positive delta G, and we've got an endergonic reaction. So stored energy. And again, simplifying this, most likely you're going to be breaking, the endergonic reactions are going to be breaking bonds. And the endergonics are going to be making bonds. Sorry for my chicken scratch, it's hard to sometimes write with my stylus on the iPad and record the video at the same time. So if you ever have questions about my handwriting and what it says, don't be afraid to ask. No judgment. All right, so we've talked about the basics of energy and chemical reactions. We've established that an individual reaction will either store or release energy. Okay, so now let's move into some concepts of, a little bit more on the concepts of reactions, and then we'll talk about specific reactions that, categories of them that we'll encounter in this course. I've mentioned that individual reactions store or release. That's true. However, in nature, in cells, most of the time, not all the time, but most of the time, reactions that release energy are going to be coupled or paired with a reaction that traps or stores energy. Okay? So that's what I mean by coupled. They're paired. So often, let's say this reaction of breaking ATP down into ADP, that's usually going to be coupled, or at least evened out with or paired with a reaction that takes that ADP and regenerates ATP. Okay? Typically, these coupled reactions are occurring in the same region of a cell. Typically, not always. And I'll leave this with your thought. Why couple reactions? Okay, what's the advantage here? Why would a cell go to the effort, I should say, of coupling reactions? What benefit would that provide to a cell, possibly? So think about that, and I'm happy to field that question in class and discuss it a little bit more. And then another concept to keep in mind, that especially as we talk about metabolism and these electron donors or electron acceptors, is this right here. Some reactions, they trap energy, but they're not going to use that energy immediately. They're going to store it and keep it, and use it use it later. Okay? Some molecules are really, really good at capturing energy, and we'll encounter them during this chapter. And I've got 3 listed here, but we're really only going to focus on these 2 here. I'm going to cross this one out for now. So this molecule called NADH and the second one called FADH2. These are electron donors or acceptors depending on their chemical state, and they are key for aerobic respiration, aka metabolism of glucose, so glycolysis, citric acid cycle, electron transport chain. If we want to maximize the energy we extract out of our food, then we need these electron donors, acceptors, to be operating efficiently in order to do that. All right, so to kind of illustrate this concept, we've got this diagram here, and it's just, there's a lot of detail here, but basically, exergonics, what I want to say about this is, if we're talking about metabolism and our cells needing to harness energy from the food, okay, so we're going to eat over here, and those molecules that we eat, those carbs, lipids, proteins, we're going to start breaking those down, breaking bonds, so that's exergonic reactions. It's going to release energy, and that energy comes in the form of these high energy electrons or energy, a little bit of detail here, but we're releasing energy, and temporarily that energy gets trapped in the form of those molecules I just mentioned, NADH, FADH2, okay? And then our cells harness the energy, the stored energy from that, to perform endergonic reactions that trap energy, okay? And in the case of aerobic respiration, the endpoint is to make ATP. So this is kind of the point. We take in molecules, we take in food, we eat it, we start breaking them down, we take the energy that's stored in there, we trap it, and we temporarily convert it, store it, and trap it in the form of endergonic reactions so that we can make ATP. So then, just kind of this last point here, ATP used for work, those three different types of cellular work that I mentioned at the beginning of this lecture. So really this kind of summary point at the bottom, the metabolic reactions that we cover later in this chapter is just a series of breaking bonds, creating bonds in order to regenerate ATP so that our cells can perform work. All right, so a couple more things on reactions. Remember that free energy change, that delta G, whether it was negative or positive. How large, so the magnitude of that change, determines whether a reaction can proceed in the opposite direction or its reversibility. You might be wondering why, but it is actually very advantageous for many reactions to be reversible. So when we say a reaction is reversible, it can proceed in the forward or reverse direction. I've already hinted at this with that concept of breaking ATP down to ADP and releasing a phosphate, but however trapping that phosphate, reforming that bond to reconvert it back into ATP. That's a reversible reaction. In fact, most of the biological reactions, so we're talking about human cells, most of the reactions that occur in our cells are reversible. Okay? Again, another question, it kind of relates to that question I asked earlier, why? What's the advantage here? Irreversible reactions, on the other hand, can only proceed in one direction. And what determines this, like I alluded to at the beginning of this slide, is that change in free energy, the delta G. The larger the delta G, the more likely it's going to be irreversible. Okay? Another concept into this, or another thing that plays into whether a reaction is reversible or not, is that activation energy. Okay? And the way to think about this is it harder to push something up a small hill or molehill versus a mountain. Which one would take more energy? And whichever one does take more energy, it's likely that that reaction would not occur in the opposite direction. So the other thing I will mention is that often the activation energy, I don't want to say is the same, but it's very similar to that delta G. So what I mean by that is the larger the E of A is more likely the larger the change in G, the free energy that's released. Okay? The smaller the activation energy, more likely is the smaller change in G. So kind of bringing this all together, we've got those two reactions here, endergonic or exergonic. Which of these two reactions is likely to be more reversible? Why? And explain. Again, I'm not going to explain it here, so think about this. We can cover it in class. I want you to discuss with each other, based on what you know so far, which of these is more likely to be reversible. The other concept here is that irreversible reactions present a problem. How do cells overcome a large activation energy or a large change of G? And really what I should say is we're mostly going to focus on that. How do cells overcome a large activation energy? All right, so that gets us through chemical reactions. That's a lot there based on your comfort and familiarity with chemistry. Now we're going to shift gears a little bit and talk about enzymes and how they assist in chemical reactions. We'll talk about 4 different types of chemical reactions, and then we're finally ready to move on to metabolism and talking about the generation of energy from the breakdown of glucose. Hopefully most of you have encountered enzymes before. If not, no big deal. We're going to start from a basic level. But the point here, when I talk about an enzyme, key takeaway here is enzymes speed up chemical reactions. They make them go faster. Okay? And the way they do this is they lower that activation energy. They help kickstart that that chemical reaction. So non-biological example that I used earlier, so lawnmower. The old lawnmowers had those pull starters or whatever you call them, those cords you have to yank on. Now we have electric lawnmowers and all that, or push button. But let's say you went to start that lawnmower and you pulled that cord, wouldn't start. Or you can only pull that cord halfway. You couldn't pull it very efficiently because it was challenging to you. Well, if you asked your friend or your sibling to come and help you and you both pulled on that cord at the same time and then the lawnmower started, well, your friend or your sibling that helped you, they would be like that enzyme. They lowered that amount of activation energy that was required on your part to get the lawnmower started in order to start using the lawnmower. That's kind of a non-biological way to think of enzymes. But the point is, they speed up the rate at which a reaction will proceed. You lower that E of A, it's going to speed up how quickly that reaction occurs, okay? Because you're talking about time as well as energy. So when we talk about these enzymes, I just want to also emphasize that the enzymes do not change themselves, okay? The enzymes just help this reaction proceed. They assist. Enzymes are also referred to as catalysts. So, maybe in your conversations with people, you're like, oh, well, this situation was a catalyst for change in our society. Okay, a catalyst is something that helps something occur. So that terminology is often applied when we're talking about enzymes. And the reason that enzymes do not change during a reaction and the way they actually help speed up reactions is often they are occurring in this manner. They bring 2 reactants or a reactant into close proximity together, and then the reaction can proceed very quickly because they're close, okay? As opposed to just hanging out on opposite parts of the cell or opposite parts of the organelle. Enzyme acts as this kind of central locator. Okay, you both are together. Now we can have this reaction proceed and we'll move on. And anything, some more terminology here, anything that an enzyme acts on, we call it a substrate. Okay, indicated right here, substrate. Substrates are reactants that enzymes act on. And enzymes, they can be activated, they can be inactivated, and they can be modulated, okay, not by the chemical reaction itself, okay, by something else. And I'll leave you with this question. I'll probably ask it again in this lecture, but why would we want to activate or deactivate or modulate an enzyme when we're talking about metabolic reactions? Okay? Why? And the simple fact is that most biological reactions or most reactions in our cells do not occur without the help of enzymes. And if they do occur, they may occur much more slowly. Okay, so enzymes really help maximize the rate at which products are produced. So here's a really blurry image, not from your textbook. I had to go out and get it online, and I just blew it up so it's blurry. But here, it's just visualizing this concept of enzymes and how they work. So we've got an enzyme here in this light blue color, and notice that the enzymes, because they're bringing the substrate here, the reactants, into the correct place, enzymes are very big biomolecules. Okay, they have to be. They generally are much larger than the substrate that they act on. Enzymes also have what's called an active site, an area where the substrate binds specifically. This is a very, and I say specifically, I really mean specifically. Enzymes are specific for their substrate. They only act on one molecule or maybe a small number of molecules that are very similar in structure. Okay, so I'm kind of going off the deep end here. But once you have an enzyme and it binds its substrate, its reactant, okay, then you have that enzyme substrate complex. The reaction occurs, and then after the reaction is finished, you have your products, okay? And then notice at the end of this, the enzyme is not changed. Its structure hasn't changed, its features haven't changed, okay? This is the visual representation of the enzyme binding its substrate, allowing for a reaction to occur. And a way we denote this in chemical form, or I should say in formula, is we have our reactants and we have our products. And notice that enzyme is not part of this, it's just facilitating the reaction, so it's often denoted above that, above the equal sign. And notice that the arrows point in both directions here, so that would indicate that this reaction is reversible. We can go from A to B, C, or on the opposite end, we can combine B and C and convert it back into A. I kind of have some words written over this, so it may be difficult to see. But if you want to think about this interaction between an enzyme and its substrate and it be very specific, I was thinking about this earlier and it made me think of my nieces and nephews that are very young right now. And they all have these toys, you know, when you're trying to teach a toddler their shapes, squares, triangles, circles. They often have these toys where you have a big block and you have these shapes, but those shapes are cut out on the block, and the child has to figure out, oh, I've got to take this square object here and it only fits into the square slot on this box, and once I do that, it goes in. Oh, I've got a triangle here, it only fits right here, it doesn't fit in the circle slot. So that's another way to think about enzymes and their substrates. All right, looking at this in a graphical format, we're going back to this, this exergonic reaction here. And the first example we gave, we had this activation energy here, and it was relatively high. And that would have been what it says right here, without the enzyme. But now we've got this reaction here. It's much lower, okay? And that is in the presence of the enzyme. Okay, so that's how we would kind of represent this on a graph. We've got our free energy on the y-axis, we've got the time on the x-axis, and you can imagine if you lower this energy, this reaction will proceed much faster, so it speeds up the reaction rate. I already answered this question. Is this endergonic or exergonic? If you don't remember, then think about that. So you might be thinking, well, what are the big deal about enzymes? We're only concerned about the reactants or the products. There's some truth to that. However, the book just provides some examples of what it's saying diagnostically important enzymes. The reality is every enzyme is important to allow for its respective chemical reaction to occur. However, in a clinical setting, we have taken advantage of the production of enzymes, and we can use them as indicators of certain typical clinical scenarios. So I don't expect you to know this, I'm just trying to provide some context for you. And the easiest one here to think about it is many of us maybe have had a relative, an older relative, grandparent, aunt or uncle that suffered a heart attack, and maybe it was like a mild heart attack and they weren't certain that the heart attack occurred. Well, they'll draw some blood and they look for what this marker of a heart attack is. And what they're looking for is the presence of creatine kinase, or CK, in the person's blood. Because when one suffers an infarction, a heart attack, okay, that causes damage to the heart, and the heart releases the enzyme in the bloodstream that normally shouldn't be there, creatine kinase, and we can detect that. So the relative amounts of CK in the blood are a good indicator of the severity of the MI that's occurred. Again, I'm not asking you all to know this, but it's just an example that there are many ways that we use enzymes diagnostically. I've mentioned this before, reaction rates are variable. Not all reaction rates are created equal. Okay? So how we measure the rate at which reactions occur, I've alluded to a few of these. So how fast the reactants disappear or how fast the products appear. Okay? And no matter what, almost every reaction can be modified by in multiple ways. So the environmental conditions. If we change the temperature of a cell, or the temperature in which a cell is existing, or if we change the pH of the solution that the cell is sitting in, so that's what we mean by environmental conditions, that may increase or decrease reaction rate. I hinted at this one, the amount of an enzyme is present. Okay, so you can imagine if you had one molecule of enzyme, it could only bind with one substrate at a time. If you had 1,000 molecules of that enzyme, it could bind 1,000 molecules of its substrate. So that would speed up the reaction. As well as the substrate concentration. So let's say you had 5 molecules of your enzyme and 5 molecules of your substrate. That's a 1-to-1, ratio. But let's say you had 5 molecules, you keep the enzyme concentration the same, 5 molecules, but you decrease the substrate concentration to 1. That's going to slow down the rate at which the reaction occurs because you're limited by the amount of substrate that's present. Okay. And I asked this question earlier. Cells do want to, for the most part, control the reaction rates. And one of the easiest ways for them to do this, because they can't really control their environmental conditions, they will typically control the amount of enzymes they are producing. Okay, so when they control the enzymes, they're either adding or removing reactants or products to impact the reaction rates. Okay, why? Why would the cell want to do this? Okay, think about energy production, think about efficiency, inefficiencies. I'm kind of giving you the answer there, but I'm happy to discuss it more in class. So, we know that reactions can be modified. Well, enzyme activity can also be modified or altered as well. Okay, and I've hinted at this, that you can activate enzymes. Okay, When an enzyme is produced by a cell, it's not immediately active. Okay, so the way I think about this is a light switch. Okay, cells produced an enzyme, but that enzyme is floating around. It's in the off formation. It's not on. Okay. However, at a certain point, that enzyme, and I'm not worried about the details of why that you know it, but that enzyme can be activated, aka that light switch gets turned on, and then all of a sudden the enzyme is active. It can bind its substrate, and reactions can proceed. So that's one way that cells can activate an enzyme. Okay. The other way is by controlling, or I should say the presence of or lack of presence of, these things called cofactors or coenzymes. Okay? So in addition to enzymes being present, most metabolic reactions also require the presence of cofactors or coenzymes. Again, they assist in the chemical reaction. Vitamins and minerals, we've all been told, oh, you need to eat your fruits and veggies so you get all your vitamins and minerals. This isn't the only reason we need vitamins and minerals, but oftentimes vitamins and minerals are assisting in metabolic reactions. They're critically important for them, in fact. So when you take in a meal and you've taken in carbohydrates and you want to metabolize that carbohydrates, aka glucose, to get energy, well, you need the presence of these certain B vitamins to help maximize the amount of energy you can extract out of glucose. If you had reduced amounts of B vitamins, then you're going to reduce the amount of energy that's present. Okay, that's an example of a vitamin. B vitamins or vitamins, obviously. Magnesium is a coenzyme; it's a metal, but magnesium is needed for glycolysis as well. Okay. On the other hand, there are ways to inactivate enzymes, to slow them down or to inactivate them completely. Chemicals can do this. So what I mean by chemicals are really either chemicals produced by the cell itself, but often external chemicals. You introduce a chemical into an environment, it can impact the activity of an enzyme. You can change the temperature, just like, so increasing or decreasing the temperature in which environment, of the environment that the cell is in, will most likely speed up or slow down the activity of an enzyme itself. And again, a change in pH will activate, or sorry, can slow down the activity of an enzyme. Okay, so this graph here is just illustrating that, and it's using pH as an example, but it's illustrating this concept here. Most enzymes in our cells, they have an optimal activity range, meaning for whatever factor we're discussing, in this case it's pH, it means in this cell, or in the human body, at a pH of roughly 7.4, you've got, over here on the y-axis, the rate of enzyme activity, and it's highest when the pH is 7.4. But if you decrease the pH, okay, it drops below 7.4, notice what happens to this rate of enzyme activity. It starts decreasing very quickly. Same can be said when you increase the pH. Okay, you're going above 7.4. That enzyme activity starts to decrease very quickly. Okay, so we could also apply this concept to what I mentioned before. If we didn't have pH, we could use temperature. Okay, and you know, in Celsius, roughly the internal temperature of our body is 37 degrees Celsius. So once we go above 37 C, we start impacting enzymes activity. We drop below 37 C, we start impacting the rate of enzyme activity. All right, we are finally to that point of discussing the types of enzymatic reactions that you're going to see in this course over and over. There are 4 categories when we're talking about biological reactions. They are oxidation-reduction reactions, hydrolysis-dehydration reactions, addition, subtraction, exchange reactions. That's kind of a clunky one to say. And then the 4th one are ligation. You know, we're going to talk a lot about oxidation reduction. We don't talk a lot about hydrolysis ones, For nursing students, you will in nutrition for sure. And then we will also discuss addition, subtraction, exchange quite often in this class, and mostly the exchange aspect of those. So let's go through each of those very briefly, okay, just to give you a little more insight into what are some of the characteristics of these types of reactions. So the first one, oxidation reduction reactions, it's clunky to say. They're often referred to as redox reactions, so reduction, oxidation, redox. And they're the ones that we're going to talk about in this lecture and moving forward for anytime we're talking about ATP. So redox reactions, what they do, they transfer electrons between molecules. Beginning, the end of the reaction, at the end of the reaction, I should say, an electron has been transferred from one molecule to another. Okay? And when we talk about the molecule that's either gaining or losing the reaction, or the electron, there's some I want you to be aware of that this matters. Anytime a molecule gains an electron, that means it's reduced. A molecule that loses an electron is oxidized. Okay? And the reason, that may seem a little counterintuitive, but if you're gaining an electron, why is it called reduced? Well, you're gaining electrons. They're negatively charged, so the charge is decreasing, so that's why it's called a reduction reaction. Easy way to remember this is the oil rig acronym. I'm from Oklahoma, so oil and gas is a big part of our economy. So I also say oil rig. But oxidation is loss, reduction is gain. Helps you keep these two straight. All right, here's a visual representation of that. It's not from your textbook, but let's just start looking at molecule A. It's got some electrons, and at the end of a reaction, notice it's lost the electrons. They're no longer there. Oxidation is loss. Okay. On the opposite side of this, we've got molecule B here, and it's showing no electrons. And then we have a reaction that proceeds, and now we've gained electrons, 2 of them. So I shouldn't say 2 plus, it should be just plus 2 electrons. They're negatively charged. But this compound gained electrons, so it is reduced. That's redox reactions. Let's very quickly talk about the other 3, hydrolysis, dehydration reactions. You might have guessed this has to do with water. Okay, so when we're discussing these types of reactions, we're typically talking about really large biomolecules. Abstract concept here maybe to you, when we convert glycogen, which is a really large storage form of carbohydrates, and we're breaking it down into smaller carbohydrates, glucose, that's an example of a hydrolysis dehydration reaction. Or if you're taking smaller complex molecules like glucose and you're making a really large biomolecule out of it, okay, that's hydrolysis dehydration. So dehydration, obviously in this reaction, these reactants, they lose a molecule of water, at least one molecule of water. That's why it's called a dehydration reaction. I've got this example here, but on the next slide, I'll show you what this actually looks like. On the opposite end of this, when you add water to reactants and a reaction occurs, you've added water, so you're hydrating it. That's a hydrolysis reaction. Okay? So the example of taking glycogen plus water, it breaks that glycogen down into smaller molecules. That's a hydrolysis reaction. You're probably thinking this doesn't make a lot of sense, but let's just look at it very quickly. And again, this is not from your textbook. I'm just providing it as a visual aid. So let me start off backwards here. We all know what sugar is, like table sugar, right? Okay. The chemical name for that is sucrose. Okay. Have you ever taken a little bit of sugar and sprinkled it in water, or your iced tea, or maybe your hot tea or something? Well, what happens to that sugar? It dissolves, right? Okay. It's because you're taking this sucrose and you're introducing it to water, or you're adding water to the sucrose, and you're creating a hydrolysis reaction that breaks sucrose down into its individual monomers, fructose and glucose. So going from sucrose to glucose and fructose, that's a hydrolysis reaction. Okay. On the other end, you can make, not you, but table sugar can be made by joining individual monomers of glucose and fructose, okay? And in the process, a chemical bond gets formed between them, shown here, and when this chemical bond is formed, it actually results in the loss of water, so going in this direction, it's a dehydration reaction. Okay. So again, I don't expect you to recreate this, I'm just trying to provide some context to you. Okay, moving on to addition, subtraction, and exchange reactions. So when we talk about these reactions, we're talking about a functional group. So what I mean by a functional group is we're not just exchanging an electron, we're not just exchanging or adding a carbon, for example. What I mean by a functional group would be like a phosphate group or a carboxyl group. So that's, or an amino acid, that's a functional group. Okay? So you're adding something to a compound, it's an addition reaction. We don't discuss a lot of addition or subtraction reactions in this class. On the opposite side of that, when you remove a functional group, then that's a subtraction reaction. So you remove an amino group from a protein for metabolism, or amino acid, I should say, then that would be a subtraction reaction. We do talk about it a lot this semester. I don't necessarily say it's an exchange reaction every time, but when you're just transferring a functional group from one molecule to another, okay, that's an exchange reaction. This is really important for biological reactions, for metabolism, I should say, because it is this process of ADP. You're adding a functional group, it's a phosphate group, okay, to ADP to produce ATP. That's an exchange reaction. Okay. I have here that in this example, when you add a phosphate to a molecule, this is accomplished by a specific enzyme to catalyze this reaction, or a specific group of enzymes, and they're called kinases. So if you hear me say the word kinase, I want you to think phosphates, and you're most likely going to be thinking ADP or ATP, okay? Here's an example of that. We'll start down here with ADP. This is, so adenosine diphosphate. We've got a phosphate group here, we've got a phosphate group here. So 1, 2, adenosine diphosphate. Now, there's kinases, they can add, see this is what I mean by a functional group right here. This is a phosphate group. So a kinase, an enzyme, it adds phosphate group to ADP, sticks this functional group here on the end, and now we've got 1, 2, 3 phosphates, so that's adenosine triphosphate, okay? And then this reaction is reversible to go from ATP to ADP. All right, last group of reactions are ligation reactions. So joining two entire molecules together. However, why they're called reaction is they use a specific type of enzyme known as synthetases. And we do talk about this a little bit, but there's not a lot I want you to know about ligation reactions, to be honest, okay? An example of this that we'll get to very quickly is when we talk about adding CoA to an acetyl group to create a molecule known as acetyl-CoA. That is technically a ligation reaction. And here is an example of that, but I'm not really going to go into it. We've got this molecule right here. You're adding the CoA to it, and it's a specific enzyme, synthetase, that converts this, but that's about all I want you to know. All right, so here's the table that summarizes the 4 reactions, types of reactions that we just went through. So, I'm not holding you to all this information that's on the table. I'd say just refer to my individual slides and what I said in this video as to as your way to review it. All right, that gets us through enzymes. So we've established what energy is, how it's needed to perform work. We've discussed chemical reactions, some of the basics of properties of chemical reactions, how they occur, how they proceed, how they're regulated. And then we just finished discussing enzymes, their role in chemical reactions. Again, properties of enzymes and ways that cells use them to control chemical reactions. So we're finally to that point where we can discuss specifically chemical reactions as they relate to metabolism. Okay, so metabolism is a word that is used very, very broadly, okay? And in fact, what metabolism really means when we talk about the definition of it is it's referring to all chemical reactions that take place in an organism. So you've probably heard people say, oh, I have a really fast metabolism, and there's some truth to that, in terms of the specifics of if we're talking about energy breakdown. But really, it's not a great term to use, okay? So talking about all chemical reactions, and that can mean the reactions that extract energy from molecules or the reactions that store energy from molecules. So in the terms of human body and our cells, we extract energy from only four, but really three macromolecules: carbs, lipids, and proteins. Those are the main source of our energy. We can extract energy from alcohol, but of course I'm not going to cover that. Okay, the other side of metabolism is the rate at which we synthesize or break down molecules. Okay, so when we talk about these, whether we're breaking down or synthesizing molecules, there's some additional terminology that we need to establish. So when we are breaking down large molecules into smaller molecules and releasing energy in that process, that is a catabolic reaction. On the other side of this is an anabolic reaction. These are reactions where we are storing energy. We're synthesizing larger biomolecules or molecules from smaller molecules. Okay, I have here as an example of this, making muscle proteins from amino acids. Okay, so the way I keep these two terms organized in my mind is that I see the word anabolic and I immediately associate that with anabolic steroids or people who are trying to build muscle. And so that's why I use the example of taking amino acids in order to build muscle protein in terms of anabolic reactions. But the point here is that the combination of anabolism or anabolic reactions and catabolic reactions result in one's overall metabolism. Cells really, really, really want to regulate metabolic pathways, whether they're catabolic or anabolic. It's critical to a cell for most cells to do this. Okay? So again, we're going to cover ways that they do this, and some of it's going to overlap with what we've established previously in regulating reaction rates and regulating enzymes. Okay? So they control enzyme concentrations. We've talked about that. You can increase or decrease the concentration of enzyme. That's going to regulate how fast a reaction takes place. Cells can also produce modulators that change the reaction rates, and most likely they're going to change, those modulators are going to change the enzyme. They're going to impact the enzyme itself. Okay? Another way that cells control metabolism is they can produce two different enzymes to catalyze reversible reactions. So what I mean by this is, let me draw it up here, enzyme number 1 creates the forward reaction, whereas enzyme 2 creates the reverse reaction. Sorry, that's some bad drawing there. But point is, a specific enzyme allows the reaction to occur in the forward direction, whereas a different enzyme allows the reaction to occur in the opposite direction. That's a form of control. Another way that cells regulate metabolism is they compartmentalize those enzymes to specific regions in their cell. And the best example of this is intracellular organelles. Okay, so an example would be our good friend the mitochondria. Mitochondria are where all the enzymes are located in order for the citric acid cycle to proceed. And that means that citric acid cycle can only occur there. It's a form of compartmentalization and control by the cell. The last one is that cells maintain an optimal ratio of ATP to ADP. So by maintaining an optimal amount of energy, it means they're going to, and cells have ways to sense this or detect this, it means it's going to impact whether it increases or decreases those catabolic or anabolic reactions. It's a form of control. All right, so I'll leave you with this question. Why would a cell want to regulate its metabolic reactions? Let's talk a little bit more about reversible reactions. Cells utilize reversible reactions, and they control the direction and the rate, or I'm kind of answering the question that I just asked you, but basically, via reversible reactions, cells can take advantage of that and control the direction and the rate at which overall metabolism occurs. So I mentioned this earlier, but one enzyme for the forward, but a second enzyme for the reverse reaction. So it really is just increasing the level of control that a cell has over metabolism and the production or storage of energy. Okay, here's a really complicated diagram. I'm not going to go into it. I don't expect you to know it. It's just illustrating the concept of these two different enzymes to control forward and reverse reactions. Okay, compartmentalizing enzymes that I spoke to earlier. Enzymes that are isolated to specific regions of the cell. This is, I've already mentioned this before, so I'm just going to, briefly mention it again, but all those enzymes for a TCA cycle are found within the mitochondria, so it makes sense. You don't want an enzyme over near the nucleus, but it's needed in the mitochondria or near the mitochondria. If they were spread out, it would slow down the rate at which metabolic reactions would occur. And then that concept of the ratio of ATP to ADP, you can imagine if that ratio of ATP is high, it means, the cell tells the cell, hey, we've got plenty of energy for the time being, so let's slow down the production of ATP, okay? So that's an efficiency thing. On the other side of that, if the ratio of ADP were high compared to ATP, then that would tell the cell, hey, we're low on energy and we need to ramp up some of those reactions to produce ATP. Okay, before we get into metabolism, we have to I need to reinforce that concept of ATP and the transfer of energy between AD and ATP, okay? So you probably have learned previously that ATP is the main energy molecule in a cell. It's often referred to as the energy's currency. And that's true, it's not the only one, but it is a major one for sure, and it will dominate many of our topics for this course, okay? But the reason that ATP is so dominant or so popular for a cell when it comes to energy is because those phosphate bonds, specifically the phosphodiester bonds that are housed in ATP, are really, really, really good sources of potential energy. They're high energy bonds, so they contain a lot of potential energy. And when you break those phosphodiester bonds, it releases a high amount of energy in order to perform a high amount of work. Just some detail here, 7 to 12 kcals per mole from breaking one phosphate bond. I'm not going to expect you to know that, but if you're chemically inclined and you want to know exactly how many calories per mole of ATP are being produced, it's right there. Here's another way to visualize ATP. Okay, so it's got this ribose 5-carbon sugar central unit. It's got an adenine group here. Again, I'm just showing this in very basic, in a basic illustration because I like this part over here. So the reason it's triphosphate is because it's got, and this isn't a phosphate molecule, it's a phosphate group, okay? So it has 1, 2, 3 phosphates, okay? Now, the bond between those two phosphate groups are what we call the phosphodiester bonds, really high energy, okay? So we've got one bond there, we've got a second phosphodiester bond between these two phosphates, okay? However, I want to point out that this is not a phosphodiester bond because it's between a phosphate, it's not between two phosphates, it's between a phosphate and carbon, okay? So this is not the same as this bond here and this bond here. So what that means is that, so I just kind of took that P away, so when you break that first bond on the end, you're going from ATP down to diphosphate, okay? So we've released some energy. Oh, and I didn't have it there. But we can break this bond. So typically what happens is this bond is broken to go to ADP. Cells can also break this bond to go to ADP to AMP, so that's monophosphate. However, rarely would a cell break off this third phosphate group. It just doesn't make a lot of sense because then it would have to recreate the ADP. So typically it just shuttles back and forth between ADP and ATP. Okay. Now, we're getting into metabolic pathways. So because ATP is the energy currency for our cells, there are a lot of chemical reactions in order to harness potential energy from bonds, trap it, store it temporarily, and then transfer that energy and recreate ATP so our cells can perform work. Our cells, most of our cells in our body, prefer carbohydrates, aka glucose, as their main source of energy. Okay? It's not the only source, it's just the main source. Okay? And for the rest of this chapter, we're going to focus on extracting energy just from glucose, not lipids or proteins. Okay? And as long as oxygen is present, then we're going to, or sufficiently, I should say, the cells, most cells will undergo 3 major metabolic pathways in order to extract as much energy from that one molecule glucose and produce as much ATP as possible. So 3 big pathways. One is glycolysis, the second is the citric acid cycle, and then the third is the electron transport chain. Big picture here, one glucose molecule yields about 30 to 32 ATP after that molecule, or that glucose molecule, goes through all 3 major steps. It's a range there because we just don't quite know. It's difficult to nail down exactly how much energy is produced from the oxidation of one molecule of glucose. Your book uses this as an overall guide to help you through the steps of these pathways, and I do recommend definitely recommend going through the book and reading through the production of ATP and how the book organizes it, because it might help you understand things a little bit better than my video or complement my video. I will warn that the book really does kind of gloss over some information at certain points. So again, use the book to complement what I'm going to present, but what I have in my video and what I'm asking you to know in my video is what I expect you to know, okay? All right, so another thing to point out before we begin talking about these in detail is where they're occurring in the cell. That's important to know. When we talk about glucose, the first step in breaking down glucose is glycolysis. This occurs, it's in yellow here, but it's a very small word over here, the cytosol. Okay, so it can occur anywhere in the cytosol. Okay, so it means those enzymes and intermediates that are necessary for glycolysis are present in the cytosol. However, the next 2 steps, the citric acid cycle and the electron transport chain, very specific location, and they only occur in the mitochondria of the cell. So all those enzymes, all the proteins and other things that are needed for the second and third step of ATP production from glucose are housed within the mitochondria. Okay, so keep that in mind. So let's begin with this first step, glycolysis. You may or may not have had it previously, which is fine because we're going to start from the ground up. So it is the first step in glucose metabolism. Okay? And like I said, it does occur in the cytosol. Okay? However, one thing that you need to know right now, glycolysis is an anaerobic process. So it does not require the presence of oxygen in order to occur. Overall, in terms of when glycolysis starts and then ends, glycolysis will actually use a little bit of energy, so it uses 2 molecules of ATP, but in the end it makes 4. So that's why I have here a net yield of 2 ATP. Also in the process of glycolysis, at the end it's going to produce 2 molecules of that NADH molecule. Okay, NADH is an electron donor, and I'll get to that here in a little bit. As well as glycolysis produces 2 molecules of a substance called pyruvate. Pyruvate is the intermediate that leads us on to the citric acid cycle or lactate production, but which depends on the level of oxygen. Okay, so kind of summary here. At the end of glycolysis, 1 molecule of glucose yields 2 net ATPs, 2 NADHs, and 2 pyruvates. The NADHs will be used later at the end in the electron transport chain, and pyruvate is the intermediate, and if oxygen is present in sufficient amounts, that pyruvate gets converted into acetyl-CoA, and citric acid cycle begins. If oxygen is not present, then that pyruvate gets converted into a substance called lactate. And then the other thing is there are multiple steps to glycolysis. And at every step, there's going to be an enzyme that's needed, okay? But there are 3, and what I mean by rate limiting is that it means that there are 3 rate limiting enzymes, and it means that these really control the rate at which glycolysis can proceed, okay? And they are hexokinase, or HK, phosphofructokinase, or PFK, and then the third one is pyruvate kinase, or PK. So we've got a busy screen here. We're going to walk through the steps of glycolysis, and this is showing the first half of glycolysis, so what I'll call the upper half. Okay, we've got key features up in the left side of the screen. Okay, and then as I go through this, there's going to be step-by-steps, and then there's also a key down here. The black circles are carbon, the white ones are oxygen, and the little p's are the phosphate groups. Okay. So I don't expect you to know the structures and how they're organized, but for me, I think having this alongside the words really helps me understand what's happening to the structure, which helps me understand overall what's happening during glycolysis. Okay, so we've got a molecule of glucose in the cytosol, and the very first step is once glucose enters a cell, it actually doesn't spend much time being glucose. It immediately gets converted into substance called glucose 6-phosphate, okay, with this enzyme, that very first rate-limiting enzyme, hexokinase. And notice here, hexokinase, that enzyme, so it's an enzyme that catalyzes the conversion of glucose into glucose 6-phosphate, but this process is energy demanding. We actually use our first ATP. So we're down -1 ATP at the very first step of glycolysis. So just keep that in mind. So we've added a phosphate group to that molecule of glucose. Now glucose 6-phosphate gets converted into something called fructose 6-phosphate. Okay? And I do expect you to know the enzymes here when I've listed them in red. But glucose 6-phosphate gets converted into fructose 6-phosphate. That reaction is catalyzed by the enzyme phosphoglucose isomerase. Okay, but no use of ATP, no generation of ATP in this step. Okay, so now we've got this structure here, fructose 6-phosphate. Okay, and it gets converted into fructose 1,6-bisphosphate. So we'll start with 1 phosphate here, and at the end of this next step, we'll add a second phosphate here. Okay, so when we add a phosphate, we're removing that phosphate from ATP, producing ADP, and it gets transferred here. So again, we use 1 ATP in this step. So the conversion of fructose 6-phosphate to fructose 1,6-phosphate to add that second phosphate group, it's energy demanding, so we use 1 ATP. So now we're down 2 molecules of ATP. We haven't even generated energy, we've used it. So now, basically what happens in this next series of steps is this molecule gets split down the middle, essentially, and we go from a 6-carbon structure into 2 molecules of a 3-carbon structure. Okay, so we start with fructose 1,6-phosphate, bisphosphate, and aldolase is an enzyme that converts this structure into this molecule called glyceraldehyde 3-phosphate. So we basically are cleaving this molecule, and then we're left with a 3-carbon molecule with a phosphate group on the end. Okay, and that molecule is glyceraldehyde 3-phosphate. Now you notice this step here. We also convert fructose 1,6-bisphosphate into this molecule called dihydroxyacetone phosphate, or DHAP for short. The reason, and then DHAP gets converted into a second molecule of glyceraldehyde 3-phosphate. The reason is because this step right here of breaking down fructose 1,6-bisphosphate, when it gets cleaved, we can't just cleave it into 2 exactly mirrored images of each other, 2 exact copies of glyceraldehyde 3-phosphate immediately. One of those has to be modified a little bit through an extra step. Okay, so that's why there's this kind of split step here in this process. But the point is, we've gone from 1 6-carbon structure. Now we've got, at the end of this first half of glycolysis, we've got 2 molecules of a 3-carbon structure, and that is glyceraldehyde 3-phosphate. And at this point, we are net or negative 2 ATP. So the first half of glycolysis is actually uses some energy. Okay. The second half of glycolysis, which I'm going to show right now, is where we start generating our energy and our electron donors. Okay. So now just a key point here is notice that every step has a 2 outside it. That's because, remember, we started out with 2 molecules of the glyceraldehyde. I'm not going to spell it all because it's right there, glyceraldehyde 3-phosphate. Okay, so each of those 2 molecules, they're going through this step. So hopefully that's clear. But if you have questions about why we suddenly have 2 here, okay, just let me know. But we have 1 molecule of glyceraldehyde 3-phosphate shown here. Okay, and now what happens is a second phosphate group gets transferred, okay, to glyceraldehyde 3-phosphate. Okay, and in that process, we actually generate our first, so 1 molecule of NADH, and that is an electron donor. Remember, it'll get used later in the electron transport chain. Okay, so now we've got this molecule, and it's called 1,3-bisphosphoglycerate. Okay, and here's this next step is where we generate our first molecule of energy, or ATP. So at this point, even though we just added one of those phosphates to bisphosphoglycerate, we're going to strip that phosphate away, and we're going to transfer it from a molecule, or transfer it from bisphosphoglycerate and add it to ADP, and we reconvert ADP and convert it into ATP. So during this step, we've generated our first molecule of ATP. But remember, in reality, we have 2 molecules. We started with 2 molecules of glyceraldehyde 3-phosphate. So this step occurs twice, so therefore during this step, we're generating 2 molecules of ATP. I forgot to mention the enzyme on the previous step. So glyceraldehyde 3-phosphate dehydrogenase is the enzyme that converts that previous step. Phosphoglycerate kinase, this is one of the rate-limiting enzymes. This is the enzyme that converts, allows the step that I just mentioned to proceed. Okay, so now the last few steps in glycolysis is just moving that phosphate a little bit, and again, removing that phosphate from the carbon structure. So now we've got this structure. It is a 3-phosphoglycerate, okay? And the next step just transfers that phosphate to a different carbon. It's not using any energy. It's not creating any energy. It's just mutating that phosphoglycerate, so it's a phosphoglycerate mutase enzyme that converts this reaction, or catalyzes the reaction, I should say. So when we have 2-phosphoglycerate, okay, we have our next to last step, and phosphoglycerate, with the addition of water and the enzyme enolase to catalyze it, it converts this, even though it looks similar in structure, it converts that molecule into phosphoenol pyruvate. So we're getting closer to that end product of glycolysis, pyruvate, but we're not quite there. Okay? So enolase converts this step, and then the very last step in glycolysis is basically removing this phosphate, transferring it to molecule ADP to generate ATP. Okay? And again, because we started with 2 molecules of glyceraldehyde 3-phosphate, and similar to this step where it was 2 molecules of ATP, same right here, this last step also generates 2 molecules of ATP. Okay, so now we've generated 4 molecules of ATP. Okay, we also generated electron donor, and again, I don't think I highlighted it, but similar to how this step and this step produce 2 molecules of ATP, we generate 2 molecules of NADH. Of 2 NADH. Okay, at the end of glycolysis, we're left with this 3-carbon structure, 2 of them, known as pyruvate. Okay, but we have to keep in mind that first half of glycolysis, we used a little bit of energy. Okay, we used 2 molecules of ATP. Okay, so from this, we have a positive 2, what I'll just say net, or combined ATP. So these are the products: 2 ATP, 2 NADHs, and 2 pyruvates. Simple, right? Okay, we're a third of the way done. So we finished glycolysis, we've got pyruvate, and now we've reached a branch point. Okay, if oxygen levels are low, then the cell is prevented from taking that pyruvate and shuttling it into the citric acid cycle, or the TCA cycle. And instead, what happens is we actually go through anaerobic respiration, or the generation of energy without the use of oxygen. And that pyruvate, it gets converted, this anaerobic reaction, the point is to convert pyruvate into lactate. And this anaerobic respiration is inefficient compared to aerobic respiration. At the end of glycolysis, when we ended up with lactate, remember we had a net 2 ATP. Anaerobic respiration does not generate any additional ATP. So through anaerobic respiration, we can only get 2 ATP. Remember, that's much different than the 30 to 32 ATPs generated through aerobic respiration. So that's what I mean by energy inefficient. And in fact, in addition to no new ATP being generated, the anaerobic respiration also uses 2 of those, the 2 NADHs that we produced in glycolysis, and it regenerates it for NAD, all right? So that makes probably no sense to you, okay? But this is what it looks like. So up here would have been glucose. We went through the steps of glycolysis. We have our products, okay? And this is what I mean by the branching point. If O2 is high, or oxygen, pyruvate will get converted into acetyl-CoA for citric acid cycle to occur. But if there's low O2, then this process happens. This pyruvate gets converted into lactate. And in this process, pyruvate, when it gets made into lactate, it takes those two NADHs that we made from glycolysis, okay, and it uses them, and it regenerates NAD. NAD is the substrate. It's the form that is used in glycolysis to generate that NADH. So I'm going to try to go backwards here. Okay, remember, so if we look here, NADH is an electron acceptor, and in this step, it accepts electrons to generate NADH. So really, we need NAD plus in order for glycolysis to occur so we can generate NADH. So that's what I mean, sorry about this, that's what I mean by anaerobic respiration regenerates NAD plus for glycolysis, because NAD plus, once it's made, it just goes back here and you can allow glycolysis. So really it would result, as long as there's low oxygen or not enough oxygen, you would just have this cycle occur. There's no oxygen and plenty of glucose. However, most of the time in the human body, in most of our cells, oxygen is plentiful. So once pyruvate is made from glycolysis, pyruvate moves into the mitochondria, and acetyl-CoA is produced, and then we can enter the citric acid cycle. Okay, so let's talk about the citric acid cycle. CAC, or TCA for tricarboxylic acid. You might also have learned this through what's called the Krebs cycle. So there's really 3 different ways people refer to it. I think the book uses citric acid cycle, so that's what I'll try to use. Most of the time oxygen is plentiful, so that pyruvate that's produced during glycolysis moves into the mitochondria. However, a point to note that even though oxygen levels are needed, we still don't use oxygen itself in the citric acid cycle. We actually don't use oxygen until the 3rd and final step, and that's during the electron transport chain. So pyruvate must be converted into acetyl-CoA that once it gets inside the mitochondria. Okay, and again, we'll talk about big picture here, the citric acid cycle, what's being produced before we go through the steps. So we start, let's see, just draw it up here, glycolysis. Remember, 2 pyruvates. So those will get converted into 2 molecules of acetyl-CoA. And then those 2 molecules of acetyl-CoA will each go through this citric acid cycle once, okay? And once both of those molecules move through the citric acid cycle, we end up with these products. We have 6 molecules of NADH. We have 2 molecules of something called FADH2. Okay? Together, sorry, these brackets are a little bit off. It should look like this. Okay. These are electron donors, and they are trapping energy temporarily for use in the electron transport chain. And this is actually, even though it doesn't look like it, it's a lot of potential energy. Okay, so this is a key kind of point to understand about the citric acid cycle. We still don't produce a lot of ATP during the citric acid cycle, but we do produce a lot of potential energy in the form of these electron donors. We only produce 2 molecules of ATP after both acetyl-CoAs go through citric acid cycle. And even though this isn't an energy molecule, it is a product, we've produced a little bit of carbon dioxide for the cell that it has to handle. Just like glycolysis, they're rate-limiting enzymes. Pyruvate dehydrogenase, or PDH, citrate synthase, CS, isocitrate dehydrogenase, or IDH, and alpha-ketoglutarate dehydrogenase, or KDH. All right, so we've got this intermediate step here before we get to the citric acid cycle. Pyruvate moves into the mitochondria, and there's a little bit of kind of an intermediate step that's needed. So in order to take pyruvate and convert it into acetyl-CoA, we need an enzyme to catalyze this reaction. And it's that PDH, or pyruvate dehydrogenase, that I just mentioned on the previous slide. So PDH converts, or shouldn't say converts, but catalyzes a reaction to add acetyl-CoA molecule onto pyruvate. So in the process, it allows for the creation of acetyl-CoA. And in that step as well, we've gone from a 3-carbon structure to a 2-carbon structure. One of those carbons gets removed, stripped away, and it actually gets converted into a molecule of carbon dioxide. And in this process, when we strip a carbon away, typically we're also stripping away those electrons. And in this step, we've generated 1 molecule of NADH, again, an electron donor. So this is electron donor, so think potential energy for the citric acid cycle. Okay, so PDH does quite a bit here in this step. There's a lot going on. Converts pyruvate into acetyl-CoA, generates carbon dioxide and NADH. Now, as soon as acetyl-CoA is made, it basically gets converted into that CoA gets dropped, and we're left with a molecule. It's called an acyl unit. Okay, and this is technically the entry, what I'll call the entry point for citric acid cycle. Okay, so here's our lovely citric acid cycle. Remember, we start with an acyl unit, but since we have 2 pyruvates, we generated 2 acetyl-CoA, so we in reality have 2 acyl units. So that acyl unit, it gets combined with, and it's not really clear, okay, but we have this 4-carbon molecule called oxaloacetate, okay, that's present in the mitochondria. And this enzyme, citrate synthase, or CS, it combines this 4-carbon oxaloacetate plus the 2-carbon acyl unit to make a 6-carbon structure called citrate. So it's making citrate with the use of citrate synthase. We're combining acyl unit and oxaloacetate, and we've got our first intermediate in citric acid cycle, okay? And you're probably wondering, like, well, where the hell does oxaloacetate come into play? I'll actually kind of touch on that at the end of the citric acid cycle, okay? So we've got a acyl unit. We've created this 6-carbon structure called citrate, going from citrate to isocitrate. Actually, I'm not asking you to know that enzyme there. I'm just asking you guys to know the rate-limiting enzymes in citric acid cycle. So we've got isocitrate, still a 6-carbon structure, but the process of going from isocitrate to alpha-ketoglutarate, okay, notice we're going from a 6-carbon structure to a 5-carbon structure. Anytime you're losing a carbon, like I said on the previous slide, you're going to strip that carbon out, produce a carbon dioxide. You're also going to strip an electron, and you take that electron, and it gets stuck onto an electron acceptor to make an electron donor. And in this case, it's NADH. So just to emphasize, reiterate here, isocitrate being converted into alpha-ketoglutarate, we lose a carbon, we generate an electron donor, and we generate carbon dioxide. Go from 6-carbon to 5-carbon, and the enzyme that catalyzes this is isocitrate dehydrogenase. Now we've got a 5-carbon structure, alpha-ketoglutarate. The next step is very similar. We're losing a carbon, and because of that, we're producing carbon dioxide and another electron donor. Okay? 5-carbon alpha ketoglutarate to a 4-carbon structure called succinyl-CoA. Okay, so, and the enzyme that catalyzes this is alpha ketoglutarate dehydrogenase. Okay. Now we're down at the bottom of the citric acid cycle. We've got a 4-carbon structure, succinyl-CoA, and at the bottom is where is the only place where we generate any direct energy in the form of ATP. So the process of going from succinyl-CoA, 4-carbon structure, to something similar is succinate. Okay? But this step is, again, I don't think I'm asking you to know the enzyme, but going from succinyl-CoA to succinate, we generate 1 molecule of ATP. Okay, something I've kind of, and I'll touch back on the 1 versus 2 here in a second. So now we've got succinate, 4-carbon structure, going from succinate to fumarate. Okay, again, 4-carbon structure, but notice we're going from all single bonds here to a double bond here. This process, we actually remove 2 electrons. We're transferring those electrons onto a really good electron acceptor called FAD. And in the process, we're making FADH2, which is similar to NADH. It is an electron donor, high energy electron donor. So this is a critical step in citric acid cycle. Going from fumarate, this molecule of fumarate, we add water to produce the molecule called malate. Okay, but no energy, no donors are produced here. Okay, and in the last step of citric acid cycle, we go from malate, a 4-carbon structure, and generate oxaloacetate. Okay, and in this last step, an electron is removed from malate, even though it's not shown here, and it's transferred to NAD to generate our last electron donor, NADH. Okay. So now we're left with this 4-carbon structure, oxaloacetate, at the end of citric acid cycle. So another way to think about this is oxaloacetate is never used up. It just gets recycled. It helps initiate the beginning of citric acid cycle, and at the end of citric acid cycle, we're left with oxaloacetate, and then it gets reused to start this process over and over and over as long as there's enough acyl units. Okay, so thinking about this in terms of all of its products, we have 1 acyl unit that goes through these steps, but really we have 2 of them. So at each of these steps, 2 molecules of NADH right here, 2 molecules of carbon dioxide, 2 molecules of NADH, 2 molecules of carbon dioxide, 2 molecules of ATP, 2 molecules of FADH2, and 2 molecules of NADH. Okay, so that's where we get 6 NADHs, 2 FADH2s, 2 carbon dioxides, and 2 ATPs from the previous slide. Okay. So now we've made a little bit of ATP, but we've generated through glycolysis and citric acid cycle a lot of these electron donors. Okay. So now the last step in aerobic respiration of glucose, we're going to produce a majority of the ATP through the use of these electron donors. So let's talk about the electron transport chain. Like I said, it's the final step in the aerobic ATP production. Okay, it occurs in the mitochondria, the inner mitochondrial membrane, and I'll show you that here in a second. And as I just mentioned previously, these electron donors, all the 2 NADHs from glycolysis, the 6 NADHs from citric acid cycle, the 2 FADH2 molecules, they really get used up here in this electron transport chain. And what they do is, the reason I call them, or they're called electron donors, is in the ETC, they literally give off their electrons. And these electrons flow down or flow across these proteins called the cytochromes. And the cytochromes help pump hydrogen ions, which helps power the ATP synthase molecule, which generates our ATP. Okay, you're probably like, that made no sense. Okay, and I've actually got some fun animation to help visualize what this process actually is. Okay, but kind of take home here is that from those electron donors and then giving off their electrons to the electron transport chain, we're making about 26 to 28 ATPs from them. Okay, and the other thing to take away from this is that during the electron transport chain is when our cells need oxygen. If they don't have oxygen, bad things happen, and this is why. This is why we breathe in oxygen. All right, so I've got a series of slides here. They're shown in steps, and I've got some animations. Okay, so let's just establish where we are in the cell first off. We've got the cytosol here, and all of this right here would be the mitochondria. Okay, so we've got the outer membrane here, we've got the inner mitochondrial membrane here, and here's the inside of the mitochondria, the matrix. You know, so we had glycolysis occur out here, pyruvate moved inside the cell, acetyl-CoA was generated, we had the citric acid cycle. So citric acid cycle has right here, you know, we've made those high energy electrons, and what I should say is donors. So this is where NADH and FADH2 come into play. They have stored electrons. They give off their electrons. Okay, these are the molecules I mentioned earlier. These are the cytochromes. Okay, they're a type of protein, and you'll see what they're doing here in a second. So as soon as these NADH and FADH2s are being produced, okay, they're donating, they're giving off their electrons to the cytochromes. Okay, so electrons here are shown in red in the little e's. So what happens? Oh, there it goes. They give off their electrons, and these electrons flow across the cytochromes. Okay. Woohoo, really fun there. Okay, I'm proud of my animation. Now, they're not just sliding down the cytochromes because it's fun. There's a reason behind this. Okay, as the electrons are flowing across those cytochromes, it's creating the energy, because that's kinetic energy, and the cytochromes use that kinetic energy to pump molecules of hydrogen from the matrix into the inner membrane space. Okay, so let's visualize that. So they've given off, those electrons are flowing, and then as they're flowing across, ideally the E's would be moving at the same time as the H's, but I didn't get that fancy with my animation. Okay, but those cytochromes are pumping the H's against their concentration gradient, going from low to high concentration. Okay, that's why it requires energy. But the point of this is that it's concentrating hydrogen ions in this inner membrane space. Okay, that's what I just mentioned. So this concentration gradient of hydrogen ions here in the inner membrane space, that is a form of potential energy. We really haven't discussed how gradients are forms of energy, but we will in one of our upcoming chapters. But the point here is that these hydrogens, they're going to go from an area of high to low concentration across this molecule called ATP synthase. That should be ringing some alarm bells. Anytime you synthesize something, I want you to think that's making or producing. And anytime you see an ASC on the end of a word, that tells you it's an enzyme. So this enzyme, named appropriately ATP synthase, is going to produce or synthesize ATPs. As hydrogens, skipped a step, go backwards, no, this way. Okay, so those hydrogens have flowed, they're being concentrated on the inner membrane space, Okay. One thing I didn't mention is we have to talk about oxygen here. Okay. So what happens is when those electrons flow, just electrons hanging out by themselves are damaging. They're free radicals. And what happens is those electrons, they combine with two molecules of hydrogen, that are also moving out here, that are available here, but they're combining with oxygen to form water, which is an inert substance. Water is less damaging than free radicals or free electrons, or individual molecules of hydrogen, okay? So that's what we mean when oxygen is needed for our cells, because it is what we call the terminal electron acceptor in aerobic respiration. If your cells don't have oxygen, the electron transport chain cannot occur, therefore no ATP production, therefore your cells die, and that's bad. All right, so that's why oxygen is needed. Now let's talk about the actual, I shouldn't say actual take home, but what we've been building towards for this entire lecture is the production of ATP from glucose. Okay? So these H's, now concentrated in this intermembrane space, they're going to move out here down their concentration gradient across that ATP synthase enzyme, and in the process, okay, that kinetic energy of the H's moving is powering this enzyme ATP synthase, and it physically attaches free phosphates back to ADP. So it's converting or reconverting ADP into ATP. Okay, so that's, and I like to use the term convert or reconvert. I don't like to use the word make ATP because that might make you think that it's making ATP from scratch, and it's not. It's just converting ADP, used energy, back into potential energy, ATP. Okay. And I just reemphasize that here. ATP is not created, it is regenerated. Okay. All right, so here is a diagram that kind of summarizes the products, the major products of those pathways. We've got glucose, we've got citric acid cycle, and the electron transport chain. Okay? So, and this just summarizes NADH, FADH2, ATP, and carbon dioxides at each level. Okay? During glycolysis, intermediate of going from pyruvate to acetyl-CoA, so we can then enter the citric acid cycle. Okay? And then products during the electron transport chain. And I do expect you to know this because I've mentioned this at every step of the way. This just presents it in a different kind of a table or graphical format. So end product here, we produce at the end, starting from 1 molecule of glucose, which is right here, okay, 6 molecules of water, 30 to 32 molecules of ATP, and 6 molecules of carbon dioxide are produced. All right, that is chapter 4. I know that was a lot to digest. But we established what energy is and how it's used to perform work, what type of work cells use. And we mostly talked about chemical work in this lecture. Went on to establish the basics of chemical reactions, reversible, irreversible, activation energies, free change in free energies, how enzymes aid in chemical reactions, how their cells use enzymes to regulate chemical reactions or metabolic reactions. And then finally, we established how metabolism is a combination of anabolic and catabolic reactions. And we went through the 3 steps of the aerobic respiration or the production of ATP from glucose. And that was glycolysis, citric acid cycle, and the electron transport chain. So there's a lot to take in there, and come to class prepared with as many questions as you have. We can rehash things. This is always a lot of information for students to take in over a relatively short period of time. And I want to say that this video, metabolism, we've covered how our cells extract energy from glucose, but it's not the only molecule that we extract energy from. And once we work through this together, there will be a second, there will be another video that is from chapter 22. It's a much shorter one, where I establish how our body gets energy from lipids or fats. This metabolic process is called beta oxidation. That will be in a separate PowerPoint that's available on Blackboard as well as a video, and refer to the syllabus as to when that will be covered in class. All right, so as a reminder, there are those learning objectives, and I'll see you in class.